Continuing on then with Tortora-Chapter4-Part1B, we're going to take a look at the prokaryotic cells starting outside the cell wall. Remember, the cell would have the cellular contents bounded by the cell membrane. Outside of the cell membrane would be the cell wall, so we're starting just outside of that. Something that can be, not necessarily is, but could be on some bacteria is a glycocalyx. That glycocalyx can take two different forms. It could be in the form of a capsule, which is a really organized, repeating structure that attaches to the cell wall, or it could be a slime layer, which is a loose, water-soluble, sticky substance that we found in the biofilm when we took a look at that in Chapter 1. Now, glycocalyx is singular, glycocalyces would be plural. Both capsules and slime layers protect the cell from desiccation, which means drying out. Now, in the case of a capsule, some organisms that are able to cause disease are also able to make a capsule. Having a capsule does not make something a disease organism, but if a disease-causing organism is able to produce a capsule, it is much more able to cause disease. That's because the capsule is made of chemicals that are similar to those in the host being the organism that is being attacked. And if we take the case of the human, it would be our immune system then that wouldn't be triggered by this capsule because the capsule is going to be made of familiar chemicals. So we would not attack that particular organism that is surrounded by this capsule. A good example is strep pneumoniae, which causes bacterial pneumonia. There are 3 designations for different strains of strep pneumoniae. S means that the organism makes a smooth capsule. M means the organism makes a mucoid capsule. And R indicates that the organism does not make a capsule. Only those organisms able to make either smooth or mucoid capsules are able to cause pneumonia. Those that are designated R cannot cause pneumonia. They do not have a capsule, so the organisms are recognized as foreign and attacked by the immune system. As I mentioned, slime layers, the other type of glycocalyx, is going to enable adherence. Adherence, or being able to attach to something, is very important. You have probably seen a biofilm as those green strands that are attached to rocks in a stream. That would be the slime layer that is enabling that adherence to the rock. Slime layers can also attach to heart valves or your teeth, for instance, and cause or enable a biofilm. If we take a look at what these look like, this is a special stain called a capsule stain. The organism is actually this smaller purplish substance inside here, and then you can see that it is surrounded by this capsule. Whereas a slime layer is not nice and organized the way that is, and that would be the sticky substance that would enable adherence in a biofilm and also coat the organisms and offer them protection from antibiotics, etc., as we discussed in Chapter 1. We'll take a look at biofilms other times throughout this semester as well. Other things that can be outside of the cell wall include flagella. Now, again, not all bacteria have flagella, but if they do, they would be considered motile. I want to point out that this is a hyperlink. I cannot activate it right now because I am not in slide presentation mode, but if you will go to slide presentation mode on the PowerPoint notes, you will be able to view that video on bacterial motility. Flagella in bacteria are composed of a protein called flagellin. That flagellin is also termed an H antigen, and I want you to remember that because we'll see how the H antigen can be used in bacterial identification. Flagella occur primarily on bacilli or rods, but there are a few organisms that have a coccus morphology that do have flagella. Now, the flagella in prokaryotes rotate 360 degrees and up to 100,000 revolutions per minute. That translates into 60 cell lengths per second, or about 670 miles per hour. So bacteria can really move fast. The flagellum can rotate either in a clockwise or a counterclockwise motion. When the flagellum rotates counterclockwise, a run is possible. A run is when one or more of the flagella that are on the surface of the cell all move together in the same direction, and it enables directional movement. Whereas when the flagella or flagellum, depending on whether the organism has one or more, flagella is plural, flagellum is singular. When the flagella or flagellum rotates clockwise, that causes the organism to tumble or turn direction. So that would not be directional movement when the flagella rotate clockwise. Now, the movement of the flagella, enabling the organism to either run or tumble, is influenced by something called a taxis, which is a stimulus. And that stimulus can take the form of either light, which is phototaxis, or chemicals, which is chemotaxis. Now, the organism may move toward the taxis or away from the taxis. So an organism might go toward light, or it might go away from light. It might go toward a particular chemical. It might go away from a particular chemical. But those would also be all be considered a stimuli that are going to influence the movement of the organisms. Now, here are some videos that you can take a look at. Again, those links would work once you're in slide presentation mode, and I strongly urge that you go and take a look at those videos. The flagella on bacteria take different arrangements. Atrichous is the designation if an organism does not have flagella. Notice that capital A means not, so it does not have flagella. Peri, the prefix, means around, so peritrichous organisms have flagella that cover the surface of the cell. And then some flagella are polar. These would be for the bacilli, where flagella are only at the ends, and they are either singly or in tufts, sort of like a ponytail. Lophotrichus would be a tuft from one pole. Monotrichus would be a single flagellum only at one pole, and amphitrichus would be flagella at both poles. Spirochetes are a special case as far as their flagella. They have what are termed endoflagella, which collectively form something called an axial filament, which instead of projecting from the surface of the cell is actually in the cell. And if you'll take a look at these animations, you'll see what that looks like. Also, I have some pictures here showing you peritrichous, where the flagella are around the surface of the cell. Here's monotrichous, where it is polar and there is a single flagellum from one pole. This is Lophotrichus, where the flagella arise from one pole and there are many, and Amphitrichus, where there are flagella from both poles. Here you can see the axial filament of a spirochete, and that is a more close look at the endoflagella of the spirochete. Well, the endoflagella would be in that axial filament. Other things that can be outside the cell wall include fimbria and pili. Fimbria are extensions on gram-negative bacteria. And they enable adherence, again, that means attachment, to other cells and to surfaces. And this can greatly increase their virulence. Virulence means the ability to cause disease. The motility that they enable, and they do enable a type of motility which is similar to pulling a rope hand over hand, they are not specifically for motility. Flagella are for motility. So while there is a little bit of motility enabled by fimbria, their primary function is for adherence. Fimbria also are going to function in biofilms by conducting electrical signals among cells. This is how the cells are going to communicate with one another. Depending on how many organisms are involved in a biofilm, the organisms are actually able to influence some of their genetic material relative to how many organisms are involved in the biofilm. So the fimbria enable that communication between the cells or among the cells in a biofilm. Now, pili are involved in a process called conjugation. Conjugation is the transfer of DNA from one cell to another. It is sometimes referred to as bacterial sex because there is actually a transfer of genetic material and there is cell-to-cell contact. We will study conjugation later in the semester. There is a slight amount, again, of motility enabled by pili. It's a gliding and twitching motility, but again, the primary function of a pili is for conjugation, not motility. Here you can see a picture of flagella. Notice how long the flagellum is relative to the short fimbria. Do not confuse fimbria with cilia. Cilia that you may have learned about are only on eukaryotic cells. There are structural differences between fimbria and cilia. So bacteria do not have cilia. They have instead these fimbria and flagella. Now again, I want to point out, not all bacteria have fimbria, not all bacteria have flagella. The pilus here, you can see that the two cells are definitely connected by this strand, and the cells then get pulled closer together and DNA is actually able to be transferred from one cell to another. This is a very important characteristic that bacteria are able to carry out, and we'll learn more about the significance of the pili and conjugation as we go on farther in the course. Now, periodically in the PowerPoint notes, I'll have a series of questions like this. You want to make sure as you're studying that you can answer these questions without having to go back and look at your notes. That means that you are learning the material, absorbing it, and processing it, and are able to answer questions about it. So we've looked at things that are outside the bacterial cell wall. Let's look at the cell wall itself now. The functions of the cell wall include giving the cell its structure and shape, its characteristic shape. The cell wall enables the organism to resist changes in osmotic force, those changes that are caused by the movement of water across cell membranes, and we'll look at that a little more later. Bacterial cell walls are involved in attachment, enabling the cell to resist antimicrobials, and they are also a site of attack for antimicrobials. They also anchor the flagella of those bacteria that have flagella. We can use the cell wall to help identify bacteria so we can differentiate among bacteria. And as we've already noted, eukaryotic cell walls are different from bacterial cell walls. We're going to look at that specifically in this chapter. Remember again that the bacterial cell wall is going to use a substance called peptidoglycan, and the eukaryotic cell does not. While most bacterial cell walls, while most bacteria do have a cell wall, there are a few that do not. An example is in the genus Mycoplasma. The Mycoplasma do not have a cell wall. An organism specifically known as Mycoplasma pneumoniae does not have a cell wall, but it is a bacterium. So what is the bacterial cell wall made of? As I keep saying, it's peptidoglycan. That's a complex polysaccharide that can also be called murine. The peptido portion is made up of polypeptides. Polypeptides are chains of amino acids that are joined together by a bond known as a peptide bond. And those polypeptides link sugar chains together. So notice that the sugar chains are going to be the glycan portion. In the peptido portion, we have tetrapeptide side chains. Tetra again meaning 4, so there would be 4 peptide side chains that form something called a cross bridge. The reason you want to be familiar with a cross bridge is that some of the antibiotics are going to attack the ability of the bacterium to form those cross bridges and therefore form a good solid cell wall. And that's how the antibiotics work, by disrupting that so that the organism cannot make a good cell wall and thus is able to be killed. The glycan portion, as I mentioned, is made up of carbohydrate alternating sugar molecules. Now these sugar molecules are very similar to glucose, but unfortunately they don't have a nice easy name such as glucose. They are instead called N-acetylglucosamine and N-acetylmuramic acid. The abbreviations for those are NAG and NAM. There are differences between the way bacteria make their cell wall. And based on those differences, even though they all use peptidoglycans, based on those differences, we are able to classify the bacteria into 2 broad categories, gram-positive and gram-negative. And we'll take a look at how we do that and what that means. So here's the structure of peptidoglycan. You can see here are the long strands of NAG and NAM, those polysaccharides, linked together sort of like logs if you were building a raft. And here are the tetrapeptide side chains, those 4 peptide side chains. And then there are other polypeptides that link these together. So this peptidoglycan can have layer upon layer upon layer of these polysaccharide and polypeptide rafts that are stacked on top of one another. So if we take a look at the gram-positive cell wall, organisms that are termed gram-positive make a very thick layer of peptidoglycan. That's actually what causes them to give the reaction that we term gram-positive. Within that peptidoglycan, they also incorporate teichoic acids. Some of those teichoic acids are linked to lipids, and they are therefore called lipoteichoic acids. The lipoteichoic acids anchor the peptidoglycan of the cell wall to the cell membrane. We also are able to use those to identify different organisms. Now, there is also a genus of bacterium we're going to be studying a lot in this semester called Mycobacterium. The reason Mycobacterium are important is that while they are gram-positive, we've looked at the structure of their cell wall and they are gram-positive, they put something in their cell wall called mycolic acids, and that makes them something called acid-fast. So we'll discuss that in lab, or you will discuss that in lab, but it means that the mycolic acid is a fatty substance that is going to resist the gram staining procedure and make these gram-positive organisms difficult to stain, and therefore we have to do a special acid-fast stain in order to stain them. So I'm going to stop here for Part 1B and see you in Part 1C.